Hey everyone, Mr. Jennings here. Today we're going to talk about other chromosome disorders besides non-disjunction events and how those can possibly impact the human body and the human body systems. Alright, so let's get on to it. So the other question is, what other chromosomal disorders can arise besides non-disjunction based? And you can have deletion, inversion, translocation, and duplication. Don't worry about translocation as much. What we need to understand, though, is these chromosome mutations can have great impact on the body because whole portions of chromosomes can contain genes. Way back in Unit 3, there was general mutations like substitution, deletion, insertions, where you only affected one base of a gene and you created a whole different kind of protein. By doing a chromosomal mutation, you can change all of the proteins found on the gene, and that can be massive. It can also be beneficial. So the impact can be disastrous or beneficial, but they usually have some sort of impact. Very few of these are very silent, but we will talk about them. So let's talk about the chromosomal mutations. Deletion is probably one that we see a lot because, for instance, we have a genome here, chromosome that's got A, B, C, D, E, and F. Those are sections of chromosomes. These are what we call loci. These loci can contain many different genes. So, and deletion, a whole portion of the chromosome could just be removed. Now, they can be removed for a variety of different reasons, damage, viruses, etc. There's a lot of things that can do that. Sometimes, a whole section of chromosomes can be duplicated. So, we have this again, the same gene, same chromosomes, A, B, C, D, E, F, and it can be duplicated. Actually, we see this quite a bit, and we are beneficiaries of this. For instance, one of the things we have in our body is the genes for amylase. Amylase is a gene that helps break down the sugar amylose. It's an enzyme for it. The amylase gene is useful for us because we have a sugar-rich diet. So humans have a higher amount of amylase genes. So the section of our chromosomes that code for amylase has been duplicated dozens of times, allowing us to produce more amylase, which allows us to digest more amylose and get more nutrition out of the sugary foods we eat. So that's an example of a duplication that can be beneficial. Next is an inversion. Let's say the chromosome gets all twisted up, and when it gets rearranged, it totally comes out in a weird mess. So instead of being A through F, it's like A, D, C, B, F. This is going to be pretty big impact because if certain genes cross between sections, these loci, they could have bigger changes. So we need to be aware of that. So they can go between there. Like the proteins, a code for a protein is going between A and B, all of a sudden E's in that place, that may change the sequence of the protein. It's not a clean break, so that's why it can be a big deal. Also, it affects crossing over quite a bit because if it crosses over, you may see a different kind of recombination altogether. Another one is called translocation. Now, translocation is kind of like crossing over, but they're non-homologous chromosomes. Translocation events do happen. This is how you get chromosomes that are longer or shorter than normal. Translocated chromosome mutations are not what we're going to talk about too much, but it's going to have a very big effect because all of a sudden, chromosomes that have genes will all of a sudden have genes that they normally would not have in the first place. And that may be an impact whenever it comes time for gene expression or inheritance. So if you inherit a chromosome from your father, and he has a translocated chromosome, you may be missing a vital gene, or you may have an extra copy of a gene that you don't even need. So translocations can be massive. Now the problems. Duplication. When you have a copied part of the chromosome, you have repetition. Now sometimes those are good enzymes, like we mentioned with amylase. They can also be disastrous. They could be enzymes for cell growth, and that could be a problem in terms of cancer. Deletion, you can remove parts of the chromosome that are vital, and you might need those genes found in those parts of the chromosome in order to survive. Inversion, it can mess up the order. The parts of the chromosomes are reattached in a weird way, and that can cause some weird issues with gene expression. And as I mentioned already, translocation. Now, human genetic disorders from deleterious genes. Now, deleterious genes means genes that typically reduce survivability. Some of those are inherited, contribute to disorders, and not from the number or shape of chromosomes. So they are inherited. not because the chromosomes are been mutated, not because the chromosomes have been non-disjunction event. They just have genes that can be disastrous. And these genetic disorders are going to be classified in three major categories: sex-linked, which are going to be found on the X or Y chromosome, and they can be dominant or recessive, but typically they're recessive. Recessive autosomal, doesn't say it there, but they can be autosomal recessive, which is the other 22 chromosomes in humans. They require 2 copies of the allele in order to be expressed. They are a recessive trait. And then there can be dominant disorders, autosomal dominant, and they only require 1 allele copy. Now, if these are deleterious genes, deleterious means, again, they reduce your survivability and your ability to produce offspring. More often than not, in order for them to stay within a population, usually they are recessive. The reason being is if they're dominant genes, if they are really disastrous for you to survive, typically are not going to be passed on because usually the kids of the dominant trait will die or be unable to have kids before they can pass on that gene to their offspring. So typically most major human genetic disorders are recessive, though there are a few notable dominant ones. Recessive disorders here, for example, is like sickle cell disease. You could be a carrier of sickle cell disease and not know it, and you could pass it on to your offspring. If both the mother and the father are carriers, there's a 25% chance the offspring will have full-blown sickle cell disease. Dominant disorders are a little different. So the main dominant disorder that we see in humans that we really like to talk about a lot is Huntington's disease. Huntington's disease that has a nervous system breakdown. Basically, your neurons and musculatory system just collapse. It's really bad. Thing is, though, it's not expressed until you're in the 30s. So it's one of those a few exceptions of dominant disorders that are deleterious. By very definition, they usually kill the person carrying them. This disease is not died out from the population because typically by the time they're expressing the disease, they have already had kids. So Huntington's disease is one of those. Most people who have Huntington's disease don't know it until they already had kids. And their kids won't know it until they either get a genetic test or until they're older. Huntington's disease has no carriers. Basically, you have it or you don't. For instance, in this situation here, we have a father and the mother. The father has Huntington's disease. He probably doesn't know he had it. And the mother does not have the gene at all. In this case, you're going to get a 50% probability of their offsprings having Huntington's disease, while the other 50% will have none at all. So it's a high-risk situation, and if the person is somehow homozygous for that trait, which would be very, very rare, all his kids would have it. But usually the person who only has 1 copy of Huntington's disease, they can either spontaneously mutate it into Huntington's, or they inherit it from their mother or father and not know it. The mother or father did not know they had it until later in life. Typically, a person, if their mother or father had Huntington's, they'll know to get tested because they'll know real quick. They can look that up. Sex-linked disorders are basically, for instance, here's hemophilia. This depends on the X and Y. Typically, most sex-linked disorders are going to be found on the X chromosome. As we can see here, X-linked is because X chromosomes are considerably larger than the Y, and they're usually recessive. So there's some dominant ones here, and let's talk some other ones. So for instance, X-linked recessive disorders, some examples are hemophilia, Duchenne muscular dystrophy, and red-green color blindness. Autosomal recessive disorders, these are disorders found that are autosomal, meaning they are found on the other 22 chromosomes. Albinism, sickle cell anemia, cystic fibrosis, PKU, Tay-Sachs disease, Werner syndrome. You can read all those effects there. There are a handful of autosomal dominant disorders. Polydactyly, meaning you have extra fingers and toes. Campodactyly is the inability to straighten the pinky. Not many people actually have that. And Huntington's disease, which we talked about, is a brain tissue degradation, which leads to a muscle degeneration and everything. You can have disorders, again, that are aberrations in chromosome numbers, like Down syndrome, Turner syndrome, Klinefelter syndrome. And aberrations in chromosome structure, basically, you have translocation events, or deletions, or insertions. And those are going to be like Crotochet, and Fragile X syndrome. Alright, so this here is an example of color blindness. So color blindness is sex-linked. It's carried by the mother, and typically that means red-green color blindness is more common in men. And we'll talk about that in the next video, why is that the case. But if you have trouble seeing the number 7 here, that means you may have some level of color blindness. Everybody has some degree of color blindness because your eyes are not perfect. You should see a green 7 right here on the screen. Alright, let's kind of put it together here. So, a lot of genetic disorders can be from non-disjunction events. They can be from chromosomal mutations. And sometimes they're just genes that code for proteins that could be disastrous for the body, and they can be considered deleterious. Many of these disorders are going to be inherited. Almost all of them are because they deal with your chromosomes. However, in some cases like the deleterious, they may cause premature death. The disease is not going to carry on. All right. What you're going to do for this activity, you're going to have a video. I'm going to have a separate video here. You're going to do a genetic disease research project on your own, make a little PowerPoint, and you're going to look up a bunch of these different disorders. I have about 24, and I'll talk more about that in a separate little video. But that will be the activity. You're going to research into this a little bit more as well. Okay, next time when we come back, we're going to talk about pedigrees and the blue people of Kentucky. Have a great time. I'm Mr. Jennings. Hope y'all are doing well. Have a great day.